Well, hello and welcome to your first lesson on cellular respiration. My goal today is to give you way too much information in a very brief overview, hopefully brief, and to really get you to focus in on what happens during fermentation. Now, there are 4 stages to cellular respiration. Glycolysis we'll cover in the next lesson. The oxidation of pyruvate as well as the structure of the mitochondria we'll do in the second lesson. Then we'll focus in on the Krebs cycle, and then we'll end up with the electron transport chain. Now, a lot of the things that we talked about in photosynthesis, the terminology is going to come in handy here because we've got a series of oxidation reduction reactions going along here, very similar to what you saw in the thylakoid membrane in photosynthesis. You'll see that there's oxygen involved, and there's also water being created by the oxygen. Oxygen is going to steal electrons from the end here, and it's going to become water. That is the opposite to what was going on in photolysis during photosynthesis, because in photosynthesis, the photolysis was to break up the water to release oxygen. And in the meantime, we also released a couple protons and we released electrons. So we're doing it the opposite way now. We've got oxygen who's going to steal electrons from a molecule built into this chain, and it's going to form water. We also have, in terms of the main overall equation, so that accounts for the oxygen and the water, we're going to have our main glucose molecule as well. And our goal will be slowly, over time, through tons of different enzyme catalyzed reactions, we're going to be chopping off each one of these carbons, and they're going to end up as carbon dioxides. Now, the carbon and the oxygen here are what become my carbon dioxides. The oxygen here became water, so don't be tricked by thinking that the O2 becomes directly CO2. It does not. Okay. Now, the end goal of chopping all of this off, there'll be 10 steps in the glycolysis, another step here, and then I don't know how many steps over here. But the main goal is to produce this version of energy. The reason that ATP is so great is that it's kind of like the currency that the cell uses. Every time we add on and cut off a phosphate on these ADP and ATP molecules, we're able to store and release really a heck of a lot of energy in those particular bonds. So we're going to try to make 36 to 38 of these suckers, and I'll explain as we go along why there's a range. And you have to understand, though, that if we're going to be talking about the reaction running in this direction, in the cellular respiration direction, there are a couple terms you need to understand and what makes it quite a bit different from photosynthesis. Now, photosynthesis was an endergonic reaction. That means that the energy, the light energy here, was absorbed, or we can say stored, in a glucose molecule. This is energy that wasn't part of the system before, and we've now added it in, right? Whereas cellular respiration is going to be an exergonic reaction, where energy is going to get released. Now, that can be a little bit confusing because you look at this and you see, well, I know there's energy stored in this molecule, and it looks like we capture the energy here. But the fact is that at every step, you lose a little bit of the energy, and that energy is going to be lost in the form of heat. Now, sounds like it's inefficient, but it's actually a really great way to keep your body at 37.1 degrees, which is, in general, way higher than the actual room temperature around you. Room temperature is somewhere around 21 degrees. So you're always quite a bit hotter. So you do need to be able to heat up your body and maintain your temperature. And so it is a good thing that it's not completely efficient. It's a good thing that it is an exergonic reaction. All right, moving on. So glycolysis, again, will be a different lesson. Oxidation of pyruvate will be a different lesson. Lesson 3 and then lesson 4 over there. Okay? Now, as you're going along, if you want to do well in this particular unit and really understand it, you need to follow all of the ATPs. So you need to make sure, again, learn more about this when we get into the details, but you'll need to remember the idea that glycolysis gives us a net of 2 ATP. Oxidation of pyruvate gives us no ATP, but it does give us a different energy-carrying molecule. We'll talk about that later. And then you need to know how many ATPs come out of the Krebs cycle, 2 for this guy, and that the majority of the ATP are actually produced in the electron transport chain. This guy is going to work just like the thylakoid membrane did. You're going to bounce some electrons around. It's going to pull the protons from one side of the membrane to another, and when it does that, it's going to drive chemiosmosis to allow the exact same molecule, ATP synthase. If you remember, we cranked the head on ATP synthase and it crushed together an ADP and an inorganic phosphate to give us an ATP. And so we're going to have the exact same kind of an idea there. It's going to be a little backwards driven again because oxygen has to pull electrons and cause everybody to steal them back. But again, that's a whole nother lesson. But this idea, the chemiosmosis idea that happens in here, gives us the vast majority of the ATP that we're going to create, right? So a couple made over here, a couple made over here, 32 to 34 made in this process right here. So we have two different names then for the stuff that gets made here, these two ATP and these two ATP, or all four altogether. We have a different name for how they're created than how this is created over here. So I've given you a little bit of a rundown over here, and I'll do a better job of it at another time. So first of all, SL, I've labeled this guy and this guy. SL is substrate level phosphorylation. If you remember back from our first unit, you learned that a substrate is the molecule that the enzyme is changing. Either you're an enzyme that builds stuff, so you're grabbing two different substrates and shoving them together, or you're an enzyme that breaks stuff and the one substrate gets broken into one or more pieces, okay? In this case here, substrate is going to be some molecule that has a phosphate group attached to it, right? And you're going to steal that inorganic phosphate from an existing molecule and make some ATP. So that's why it's called substrate level phosphorylation. An enzyme is doing the job and it's stealing a phosphate from a substrate that's already exists. Okay. Then we get oxidative phosphorylation. Now, oxidative phosphorylation, it's what's happening up here. It requires you to have that electron transport chain happening. It's chemiosmosis being driven by protons jumping across a membrane, right? And then flowing out through ATP synthase. So this is very different. This oxidative phosphorylation is all driven by chemiosmosis, and this is the one that requires ATP synthase. So you're going to get 4 made by substrate level phosphorylation, and then you're going to get the 32, the vast majority, made by oxidative phosphorylation. So keep those terms in mind. They'll come up again. Also on this particular diagram, just before I move on, oxidation of pyruvate is what we're going to call the stage because literally pyruvate is oxidized. Much easier to call it that. This particular web page calls it transition reaction. This particular page calls it the transition reaction, but it's basically the idea of how do you get pyruvate to become the right kind of molecule that can then jump into the Krebs cycle. And we'll go in lots of detail as we run through there. So here's another part of the brief overview on aerobic cellular respiration. Now, you have already looked up the words that refer to obligate aerobe and obligate anaerobe, and also facultative anaerobe. An aerobe or aerobic is when you do require oxygen in order to create your energy. So the aerobic cellular respiration, this is what you are doing the vast majority of time. You need oxygen, you have your mitochondria that are going to use it, and you're going to produce a lot of ATP for that. A lot of value per glucose if you're doing aerobic cellular respiration. So here's your overall picture again, your overall idea here. So again, you get a glucose, it goes through a process called glycolysis, you pump out a couple of important products, and you get yourself a pyruvate. Keep in mind, pyruvate, pyruvic acid, the difference is so ridiculously small, we're going to use them interchangeably, okay? Just the acid version versus the not acid version. So we will be using these two terms interchangeably. The same thing is going to go for lactic acid. You've heard of that before because you've just gone through your fermentation lab. Lactic acid and lactate, same deal. It's pretty much the same molecule, you're just missing an H. All right, so you're going to get the pyruvate. Keep in mind, this is where some people get screwed out. You're going to go with the pyruvate into the mitochondria before you do anything else with it. And the mitochondria actually has 2 membranes. I'm going to scroll up here for a second and you can see it again. There's this outer membrane, which is kind of like the shell of the whole thing. And then you get this inner membrane that does this weird folding thing. These folds are called crista or cristae. You'll see that in a separate note, so don't worry about it for now. But you're going to have to get that pyruvate from the outside all the way to the inside, and you're going to oxidize it in here. So keep that in mind. This picture is a little deceiving. It looks like it gets oxidized outside, but it really doesn't. It happens inside the mitochondria. All right, let's keep going. So the pyruvate then moves in, and you're basically going to strip it of a carbon dioxide and steal some electrons from it to make another molecule. And then you're going to, through a series of chemical reactions, you're going to be chopping up, and you're going to get, in the end, your 6 carbon dioxides, 34 ATP, there's the 2 plus the 34, and then 6 water at the end. So this is basically showing you an overall idea. If you were to look at your overall equation, that this is basically what's the general overall picture. So again, very briefly, we're going to do a note on this next day. Stage 1 then would be glycolysis. Glycolysis basically means you're breaking up a sugar. Glyco is the same word that we see in the word glucose. And then you know that lysis means to burst. So it's the breaking up of my glucose, right? Now, all organisms do this. All organisms. If you're going to use sugar for food, you are going to go through this process. So we're including in both prokaryotes, which are your bacteria, and eukaryotes, which is you and every other organism on Earth, fungus and plants and all those guys. All of them are going to use glycolysis. Now, the thing is, only 2 ATP are produced. So you're not doing a heck of a lot, just 2 ATP. But the nice thing is that it doesn't require any oxygen. So if you're an organism who doesn't have a very high energy requirement, 2 ATP, you might feel rich. But if you are larger, like the wonderful human specimen that you are, then you're going to need a heck of a lot more ATP per glucose. Otherwise, it'll in very short amount of time end up being a waste of your time to even try to break up glucose, right? Now, there's 10 steps. We're going to go through all 10, but we're not going to mention the name of every single enzyme, just a couple of the important ones. But there are 10 different enzymes doing the job of manipulating the bonds on a normal glucose, adding phosphates, taking them off, stealing electrons, moving little functional groups around, all that kind of stuff. You'll see those details later. Now, it is an exergonic reaction, so we will lose a bit of the energy. But the goal is take the glucose. At the end, you'll have 2 of the pyruvate molecules. Notice that we've got a 6-carbon molecule. Now we've got 2 3-carbon molecules. So we're chopping this sucker in half. And in the meantime, we are going to use a couple ATP. I know that sounds weird because our goal is to make ATP, but we have to prime the pump. So we're going to add a little bit of energy by stealing a phosphate, shoving it onto the glucose molecule, just so we can destabilize it and help it to fall apart, help the enzymes do their job. But then in the end, we're going to steal some electrons. If you remember from photosynthesis, you had a molecule NADP. Well, now instead of nicotinamide adenine dinucleotide phosphate, we're using this sucker, just NAD. That's all you need to really know, NAD. And we're going to do the same idea. If you give it the hydrogen, remember the gift is really the electron. So the NAD is going to become NADH. And I have a couple extra inorganic phosphates floating around. And I'm going to end up using those. And in the very end, you're going to see, again, this will all be details tomorrow, we're going to end up with creating 4 ATP. So we used 2, but we made 4. So that means by the end of the process, we have a net of 2. We're up by 2 ATP that we didn't have before. Prime the pump, but we made more than we started with. Okay, so keep that in mind. Although glycolysis, you do make 4, you're going to get a net of 2. Okay, so the main stage is glycolysis. You don't need oxygen for it. Everybody does it. Very limited yield. Now, let's supposing, again, you're a big, wonderful human being specimen. You also have the ability, because you have a mitochondria, you have the ability to go through oxidative respiration as well. And again, a whole bunch of exergonic reactions, so heat will be produced, but we're going to try to take these pyruvates, the ones that you just created up here, take the pyruvate, and through a series of reactions, oxygen will be involved here, we're going to end up making a heck of ATP and end up with our final products that you see in the main equation. I'm not going to say much more than that on this because that's not the focus of today's lesson. I'll teach that again another day. Just as a point of information, just for the heck of it, I want to remind you, I think I told you this before, that mitochondria have their own DNA. In fact, when you sequence the DNA of the mitochondria, they found there are 37 different genes. What's interesting about mitochondria is that you got every single one of your mitochondria from your mother. Your father did not contribute any. Your father gave you some nuclear DNA, but he didn't give you any of the cell parts. All of your cell parts come from your mom. So every human on Earth has the DNA, which is the nuclear DNA, pardon me, which is a mix of their mother and their father, but they have only their mother's mitochondria DNA. So that gives you an opportunity to actually map families in a little bit of a different way. So 37 different genes, and you can compare the mutations in these genes between people to see how closely related they are. There's a lot of really interesting research going on looking at the differences and similarities between species types. The other interesting thing is because they have the genes, like why bother having these genes? It's so that they don't have to rely completely on the nuclear DNA or the production of proteins from the nucleus to run all of their programming. So they can replicate on their own. They have all the information they need to double themselves up, so they don't rely on the nucleus for that. And they also produce their own proteins. So any enzymes that they're using, 13 of them, they're going to produce their own and not have to worry about it. Okay, so here is the actual real main part of what today is. The rest of it was just a preamble to get you ready for oxidative respiration, which we will talk about, or aerobic respiration, which we will talk about for the rest of our unit. But I need to highlight this right now. This is the anaerobic cellular respiration, right? Anaerobic. So we know aero, not oxygen, not requiring oxygen, all right? Now, glycolysis on its own is actually considered anaerobic. There's no oxygen involved. But we're not talking about anaerobic in that sense. We're talking about anaerobic in the sense that once you get the pyruvate, what do you do with it? All right. Now, if it is aerobic cellular respiration, then if you have oxygen, you take the pyruvate and you go through a series of reactions and you end up with these goodies right here. You end up with the 6-carbon dioxide and the 6H2O through a whole lot of steps that you'll see later. But the pyruvate itself, this dude right here, if I don't have oxygen available, or I never have oxygen available because maybe I'm an obligate anaerobe, then I'm going to go through a little bit of a different process. The first type of fermentation I want to talk to you about is ethanol fermentation. Simply called that because the end product, the end here, is going to be an alcohol known as ethanol. The OL tells you it's an alcohol, and that's also why we are going to end up calling this alcoholic fermentation. So if you're doing some of your research, answering questions, you may see it called alcoholic. You may also see it called microbial. Microbial simply because small things like single-celled yeast are able to go through this. Now, there are some bigger things that are able to go through ethanol fermentation. Your goldfish would be one of them, things like that. There's a few different fish that can do it. Now, why would yeast, who is not actually a bacteria, it's actually a fungus, which is eukaryotic, why would they ever want to go through this process, the pyruvate and whatnot? Well, it's simply because sometimes yeast run out of oxygen. If you throw yeast into a barrel and you shut it off and they run out of oxygen, they're going to start producing alcohol for you. And that's exactly how we get the alcohol in wine and beer. You throw some yeast in there, they munch away on the sugar that you give them. They end up going through glycolysis quite successfully. Once they have all their pyruvates, because they cannot go through oxidative respiration, the oxygen runs out, they're going to go through ethanol fermentation instead. Okay. Now, you might be wondering, where does the energy come from in all this? Because this whole section here, the whole fermentation, actually doesn't produce any ATP whatsoever. So I hope you're asking the question, what's the point? If we're not getting any energy out of it, why do we care? Well, if you notice that with the glycolysis, when we take the glucose and we end up chopping it into the 2 pyruvates, we get a net of 2 ATP, and we also get a couple of these NADHs, right? And that's going to become more apparent what we're doing with these. These are basically the high energy electron carriers. So I've made my ATP, but the thing is, if I end up with an NADH, I'm not going to be able over time to continue running through glycolysis and get this limited bit of energy, because I'm going to run out of free NADs. So my goal here from the pyruvate to the ethanol is to restore the amount of NADs I have available so that glycolysis can keep running. You don't want glycolysis to stop, because even though it's a tiny bit of energy, it's still more energy than you had before. So it's still a slight benefit to an organism who is used to having lots of oxygen available and now has none. And so that's why we're doing this whole extra step here. I want to point out the name as well of this enzyme. Notice it's called pyruvate decarboxylase. I want to remind you that a carboxyl group is basically a CO2. It's a carbon, C with a couple O's on it and sometimes with an H on it. But the way that these guys are arranged, this is basically the carboxyl. So basically what we're doing is we are taking the pyruvate and chopping off a carbon so that we end up with a 2-carbon molecule. And in the process of doing that, a couple of these oxygens are going to go with it, and I end up with a carbon dioxide. So pyruvate D to remove from carboxylase, the enzyme that removes a carboxyl group from pyruvate. All these names are based on exactly what these things are doing. So hopefully if you can put the pieces of language together, it won't be too tough. So where do we use this? Well, we talked about this back when we talked about enzymes in the first unit, but they are used when we're making bread. This is the alcohol is what's going to give flavor and whatnot to, and actually the alcohol itself, to beverages. But the carbon dioxide is going to produce bubbles, and that's going to help your bread rise. Okay, so in winemaking, you've got that max ethanol that can be produced then is 12%, because when you let this process run, eventually the ethanol builds up to such a high level, the 12%, that it becomes toxic to the little yeast that are performing this chemical reaction. So when you start to kill off your yeast, you're going to get to a max amount of ethanol that can be produced, and the yeast cells are going to die. Okay. This is also why plants that get overwatered, why some of them, although we do have aquatic plants, but the ones who aren't normally aquatic, if you overwater some of these land plants, they will actually drown because they don't have this system of being able to go through a fermentation to at least keep glycolysis going. So if they get to this point here and they make the pyruvate and they can't do anything with it, glycolysis shuts down because you can't replace the NADs, and then they're going to end up literally drowning. So don't overwater your plants. You got to learn what type of species like what type of water, okay? Because they don't have this ability to run through this separate program. So this is what, again, your goldfish and your fungus will do. Some of your yeast cells will do. You, on the other hand, and this is the pain that you felt when you guys were doing the lab with the little clothespin. Hopefully you got that done. If you haven't, please go back and get that done. The lactic acid fermentation, obviously called lactic acid because that's our final product here, okay? Please notice there's a few little weird things happening here. So we have our normal glycolysis. Again, this is not part of the actual fermentation. The boxed out sections are the actual fermentation section. So we end up go through glycolysis, we make a couple NADHs, we make our 2 ATP, got a little bit of energy there. And of course, we're doing this because we've run out of oxygen. And we're going to have our pyruvate. And instead of taking it into a mitochondria where I can make tons and tons of ATP, I'm going to run it with this called the lactate dehydrogenase. Dehydrogenase is something that removes hydrogens from something. But if you notice here, I've got 4 hydrogens and 6 hydrogens. So I haven't removed hydrogens from this sucker, except that I actually really have. I have removed them from the NADH, and I've shoved them on. So there's 2 NADHs. I've stolen that, and I've given it, and I've created the lactic acid. And part of the reason why it's called lactate dehydrogenase is because it's the same enzyme that allows me to reverse when I go back the other way. So as soon as oxygen is available again, I'm going to take the lactic acid, I'm going to reverse it back the other way. And I'm going to get my pyruvate, take it into my mitochondria, and business as usual. So again, we have the idea here that if I'm out of oxygen and I have to do glycolysis here, I need to have a supply of NAD to make the reactions work, to make the NADH, and that's why I have to take the pyruvate and do something with it so I can get the NADs back, so this can keep happening. And if this keeps happening, then I'm going to keep getting more and more ATP. Very little ATP, but at least it's something. Now, lactic acid has been blamed for all sorts of stuff, but it is that feeling that you get after you've had a big workout. You know how it's a little bit sore first day and then the next day you can barely walk? Well, they used to say that lactic acid causes all of the stiffness, all of the soreness, and all of the fatigue. But as they've learned more and more about what's actually happening, They realized that some of the soreness is actually caused by the fact that whenever you move your muscles excessively, you're actually ripping the muscle fibers. So it's actual pain you're feeling is actual pain. It's from broken tissue that's being restored. There has been less information saying that it's stiffness. They used to think that the lactate molecules would form crystals inside your cells and do all sorts of silly stuff. So it's not so much about the stiffness anymore. Yeah, for sure, there's still something attributed to the soreness and perhaps the fatigue. There's a lot more research being done in this particular area to see what's really causing this problem with muscles. All right. Now, again, the lactic acid can be converted back into pyruvate when O2 becomes available again. When you are out of oxygen, and when lactic acid fermentation is triggered, it's because you have something called an oxygen debt. That refers to all the O2 that you need to try to reverse this process and get you back to doing normal oxidative respiration. Okay, so oxygen debt, key term, make sure you know what that means. It's when you don't have enough oxygen to run normal processes and you have to go through lactic acid fermentation. Okay, I'm not going to belabor this little point. I don't even really love this diagram anymore. So I'll let you take a look and read through this on your own. But again, the basic idea is glucose to pyruvate. I'm going to create a couple NADHs and create a couple ATPs. That got wiped off there. Make sure you know that that says ATP, not ATI. Alrighty, and that's that. And same thing here, ethanol fermentation. Go through normal glycolysis, creating a couple ATP, a couple NADH, and I get them back when I convert the pyruvate to the ethanol. There is this other in-between step that I didn't put on the last diagram. So pyruvate, after you chop off the CO2, it actually ends up being something called an acetaldehyde. Acetyl is talking about the two carbons here. And then you go through another process where you are going to add a couple of those hydrogens and make your ethanol. Okay, so a two-step process, but I'm okay with if you just know this version of it. That's totally fine if you just run through and say, this is what happens. It doesn't matter that there's an extra step in here. It's too much information for grade 12. So here's our big overview then, comparing photosynthesis to aerobic cellular respiration. So not the fermentation that I've just finished talking about, but just your overall general, when you do have oxygen available, aerobic cellular respiration. So start off with your chloroplast. Remember, you've got your thylakoid discs, the lumen on the inside. You got the jelly here, which is the stroma, and then you get the inner and the outer membranes, right? Sunlight strikes the photosystem 2. You get the photolysis of water happening, water in, O2 out, along with a couple of electrons that bounce through the system. And as they're bouncing through the system, remember that they end up with the end goal to reduce NADP, notice P, think P for photosynthesis, NADP into NADPH through that reduction. Got that nice, beautiful, fancy word, okay? And you know that at the same time as those electrons are bouncing through the membrane, you are pumping protons into the lumen. And you are going to use them to drive chemiosmosis, and that will phosphorylate ADP into ATP. And then remember that the ATP and the NADPH, these high energy electrons, get delivered to the Calvin cycle, and the ATP as well is going to give up its phosphate, and they're going to help to change rubip and carbon dioxide and to move and rearrange the molecules once you shove these two together to end up with your G3P. Then, of course, most of the G3P go back and make more rubip, but one of them becomes a half glucose. So glucose is our end goal there. Now we're jumping into the cellular respiration story, because now we're going to take glucose and I'm going to, in 10 steps, chop it, move some stuff around, make some ATP, make a couple NADHs, and end up with a couple pyruvates. I've gone from a 6-carbon molecule to 2 3-carbon molecules. Then the next stage will be to oxidize that. That means I'm going to steal electrons, oxidize. I'm going to steal electrons from the pyruvate. And while I'm doing that, I end up chopping off a CO2. It says 2 here, 2 CO2, because there's 2 pyruvates. So 1 CO2 per pyruvate and 1 NADH made per pyruvate, right? The end molecule is going to be called acetyl-CoA. Acetyl-CoA enters the Krebs cycle. Whole bunch of things, just like we had over here where we already had rubip and we were crunching it together when we fixed the carbon dioxide. Same thing here. The acetyl, which is bringing 2 carbons with it, is going to be crunched together with another molecule. I'll give you all their beautiful names later. And in the process, I'm going to run through, and in the process, I'm going to end up producing a couple ATP. Now, again, it's a 2 because there's 2 pyruvate. This cycle has to run twice. So 2 times for the Krebs cycle. And then the beautiful things that after running twice the Krebs, I'm going to get directly a couple of ATP. But I'm also going to produce a lot more of these NADH molecules, and in addition, a few of these FADH2 molecules. This is a high energy electron carrier. This one's high energy, but it's just a little less high than this sucker right here. But they both are going to carry electrons from the molecules in the Krebs cycle, and they're going to donate them to the electron transport chain. This is going to make them bounce through a membrane just like what happened over here. So you have electron transport chain just like you had over here in photosynthesis. And by doing that, you're going to pump protons, drive chemiosmosis, turn the head on a beautiful ATP synthase, and crank out a ton of ATP. Now again, this part is what we call oxidative phosphorylation. So oxidative phosphorylation, because of the redox reactions that are happening here. And it just happens to be that the very end product that you get here is the oxygen over here is going to steal the electrons and become water. So you can see that between these 2 systems here, the electron transport chain, whether it's photosynthesis, producing oxygen and using H2O, or whether it's cellular respiration, using the O2 and producing the CO2, these guys are back and forth. And the same thing with this. The carbon forms a cycle down here between the Calvin and the Krebs. Calvin, Krebs, Calvin, Krebs. So between the sugars and the carbon dioxides. So this is how we've always told you that you produce CO2, plants use it to make O2. Well, sort of. The carbons are in their own cycle. The oxygen is in its own separate cycle. But certainly the O2 that is being overproduced by the photolysis of water is definitely of a benefit to cellular respiration. So that is that there, and some more details here for you to go through. This is a very good comparison for where things are happening, what are your major players, and what type of electron carriers you have. So we've already covered that. Some more information here. Again, we had a couple of stages. You had the light stage, and then you had the light independent stage, or we can call it photostage, synthesis stage. This is where you had photons of light exciting the chlorophyll, and we end up splitting water into its parts. That all happens in the thylakoid membrane and the lumen. And then once we've produced our NADPH, NADP for photosynthesis H, and a couple ATP, we use them to help convert the CO2 into the G3P during the Calvin cycle. And that, of course, happens in the stroma. Okay, and then eventually, we skipped this whole conversation, but eventually G3P is going to become a glucose molecule. Okay, so again, this is photosynthesis as a quick overview. Then aerobic cellular respiration, again with its 4 parts: glycolysis, oxidation of pyruvate, Krebs cycle, and the ETC. The glycolysis is going to produce a couple of ATP through substrate level phosphorylation. Basically, you want to start off with 1 glucose and end up with 2 pyruvates. It happens in the cytoplasm. But then this little section here has a couple of things. First, I need to take that pyruvate that I've just made here, and I need to strip a couple of electrons off of it. And during that stripping and stealing those electrons, I end up with an acetyl-CoA and a couple CO2s. Then I take that acetyl-CoA and I chop. I shove it together with a bunch of other molecules. And in the end, the 2 carbons per acetyl, which gives me a total of 4 carbons, 2 times 2, end up here as the 4 carbon dioxides. So by the time this is done, we've got 2 carbon dioxides chopped off of the pyruvates, and the final 4 here. These are all 6 of the carbon dioxides that belong, where is it down here, in the overall equation right here. Okay, so you get your glucose in, and you follow the carbons out. Okay, glucose in, carbons out. And then you get your 6O2 in, stealing electrons and forming water. 6O2 in, 6H2O out. Okay, you're going to have to learn specifically where these things happen, just like you did for the photosynthesis unit. And of course, the end goal is that by doing all of this, manipulating of these molecules, we want to have our 36 to 38 ATP, 2 of those being produced during glycolysis. All right, and that, my friends, is the end of today's note. Good luck with all the rest of your studying, and let me know if you have any questions.